Hi everyone, welcome back to Science Simplified. Today, we have an exciting paper to discuss about photosynthetic artificial organelles. It's all about creating tiny energy factories inside artificial cells.
Yeah, it's like building a mini solar panel in a bubble. But first, let's introduce ourselves. I'm Itai, your go-to guy for overly complicated analogies.
And I'm Eden, here to make sure we don't get too lost in the science language. Before we dive into the science, let me introduce the paper we are discussing today. It's titled Photosynthetic Artificial Organelles Sustain and Control ATP-Dependent Reactions in a Protocellular System. And it's authored by an international team including Kil-Young Lee, Sung-Jin Park, and others.
Thank you, Eden. Now, let's break down what all that means.
So Matan, what's the big deal with these photosynthetic organelles?
Well, imagine a tiny bubble, like a soap bubble, but this one is packed with super cool machinery. Inside this bubble, we have two photoconverters and an ATP synthase. These are basically tiny machines that convert light into energy.
Right, and these photoconverters are like two different brands of solar panels. One is from plants, photosystem 2, and the other is from bacteria, Proteorhodopsin. They both help make ATP, the energy currency of the cell.
So let me get this straight. They use light to create energy, and it is possible to control this process?
Exactly. Using different light wavelengths, we can remote control the cell's energy production. But we will talk about it later. They encapsulate these organelles inside giant vesicles, which are like big bubbles, to create a protocellular system.
Oh, and here's the cool part. They tested this system with two ATP-dependent reactions. One is carbon fixation, and the other is actin polymerization, which helps cells move and change shape.
So this vesicle can actually change shape and grow actin filaments, kind of like our muscles in our bodies. That's wild.
Absolutely. They even showed that the system could sustain ATP production for days and control actin polymerization inside the vesicles. This could pave the way for developing more complex artificial cells that mimic real cellular behavior.
Imagine one day we could have fully functional artificial cells that can repair themselves, adapt to changes, and maybe even produce their own energy just like real cells.
And all of this starts with these tiny photosynthetic organelles. It's like the first step towards building tiny, self-sustaining robots. The future is bright, literally, with all this light-powered tech.
Now, before we dive deeper, let's talk a bit about something central to this whole process, the proton gradient. Itai, want to take this one?
Absolutely. So, the proton gradient is like unsung hero of ATP synthesis. Imagine you have got a dam holding back water. The water wants to flow through, and the energy can be harnessed. In cells, this water is actually protons, or H plus ions, and the dam is the membrane.
Right, and these protons get pumped across the membrane, creating a high concentration on one side and a low concentration on the other. This difference in concentration is what we call the proton gradient.
Exactly. ATP synthase, which is like a tiny molecular turbine, taps into the stored energy. The protons flow back across the membrane through ATP synthase, and this flow drives the conversion of ADP and phosphate into ATP, the cell's main energy currency.
So, in simpler terms, the proton gradient is what powers the ATP production. Without it, the whole process would grind to a halt. And in the paper we are discussing, they created this proton gradient using light. It's a neat trick that mimics what happens in real cells, but in an artificial system.
Speaking of light, Let's dive a bit deeper into how these photoconverters work. Photosystem 2 from plants uses blue and red light to kickstart the electron transport chain, creating a proton motive force that drive ATP synthesis.
And Proteorhodopsin, the bacterial counterpart, mainly uses green light for proton pumping. What's fascinating is that at different pH levels, Proteorhodopsin can pump protons in different directions, adding another layer of control.
So with these two working together, we can finally tune the energy production in these artificial cells. Red light to boost it, green light to slow it down. It's like having a high-tech dimmer switch for cellular energy.
As I mentioned earlier, they coupled this energy production to other cellular processes. For instance, they use the ATP to drive actin polymerization, as shown in Figure 4c and Figure 4d. When exposing the vesicles to red light, the production rate of actin has significantly increased 0.22 ppaa, percent of projected actin area divided by minutes, in compare to green light exposure that has shown almost none, 0 ppaa per minute.
Right, and the actin polymerization is a key process in real cells. It helps with movement, division, and maintaining cells' shape. By controlling this process, they are mimicking some fundamental aspects of cellular behavior.
And don't forget about carbon fixation. They demonstrated that the ATP produced could be used to convert pyruvate into oxaloacetate, which is a critical step in many metabolic pathways.
And get this, the team used both fluorometric assay and GCMS to measure the production of oxaloacetate. The results showed a significant increase in oxaloacetate concentration under light stimulation, confirming that their system was effectively coupled ATP synthesis to carbon fixation.
To put some number on it, they found that the concentration of oxaloacetate foam was significantly higher when the artificial organelles were illuminated, compared to when they weren't. This demonstrates that the ATP they were producing was being used directly in the carbon fixation process.
It's like they are building the foundation for a tiny, light-powered factory. This could lead to all sorts of applications, from bioengineering to energy production.
Imagine artificial cells that can produce their own energy, fix carbon, and even change shape in response to their environment. The possibilities are endless.
And all of this research brings us one step closer to creating fully functional artificial cells. Cells that can mimic the complexity and functionality of real living cells.
Who knows? Maybe one day we'll have artificial cells that can repair tissues, produce medicines, or even clean up environmental pollutant.
Well, that's all the time that we have for today. Thanks for joining us on the deep dive into the world of artificial photosynthesis. Remember, science can be complex, but here at Science Simplified, we make it easy to understand.
And fun. Don't forget fun.
Definitely. See you next time and stay curious.
Facts and figures guide, making knowledge grow.